In today's lesson, we will look at the type of chemical bonding present in amino acids, then the intermolecular forces that are likely to exist between amino acids and other molecules, and then the actual bonding that takes place between the amino acids themselves. So the amide bond, also known as the peptide bond when studying biology particularly. Let's first of all begin with the general structure of an amino acid. And it has a central carbon which is bonded to a hydrogen. Then on one side it has a carboxylic acid group, and then on the other side it has an amino group, and then an R group, right? Now the R group is something like, it could be a CH3, it could be a alcohol group attached to an alkyl part, so maybe CH2OH, and so on. So there's 20 different naturally occurring amino acids, and they all have different R groups. So that is the general structure there. And one other point worth mentioning is, you can see from this picture here, if the R group isn't hydrogen, then this carbon here, also known as the alpha carbon, is chiral, which means it has 4 different substituents attached. So let's address the first question. Types of bonding present. All of these bonds between the carbon and the oxygen and the hydrogen, they are covalent bonds. Single covalent bonds, we've got a double covalent bond there with the acetyl bond. Then if the amino acid is in the solid form, also known as the zwitterion form, where this lone pair on the amino group on the left takes off the hydrogen on the right, it gives this structure. So this is known as the zwitterion and exists in the solid form. And we can simplify that diagram to just think of it as like a rectangle with one side being positive and the other side being negative. And then another amino acid can join side by side, positive, negative, and you get this positive, negative, positive arrangement. Okay, so you have ionic bonding when the amino acid is in the zwitterion form, and then that leads to, powdered amino acids having very high melting points, as in like over 400 degrees C in some cases. So we got covalent bonding in the standard molecule, and then ionic bonding in that zwitterion form. So that's the first part addressed. The next thing is to look at the intermolecular forces. So the intermolecular forces depend on the different parts of the amino acid. So the carboxylic acid part, and indeed the amino bit on the other end, they both have lone pairs. So both of these groups are capable of hydrogen bonding. So hydrogen bonding is the strongest intermolecular force, and one of the strongest intermolecular forces, so H bonds there. Then, as with any molecule that has permanent dipoles, so let's just draw one obvious one there. So that would be, it has permanent dipoles throughout the molecule. The other intermolecular force present is therefore going to be permanent dipole. And then, common to all molecules, whether they have polar groups or non-polar groups, is the presence of van der Waals. So van der Waals forces, or London forces as they're sometimes referred to, is the temporary attraction between two molecules involving instantaneous dipoles, induced dipoles. So IDID, instantaneous dipole induced dipole forces. And they are particularly prevalent when you have long carbon chains. So if this R group was, say, a long carbon chain or had a benzene ring, so the presence of carbon and hydrogen with lots of electrons, then that gives rise to stronger van der Waals forces. So we can see the amino acid is capable of all 3 intermolecular forces, and depending on what it's bonding with, that will determine which force is dominant, which one is the one to talk about. The next discussion involves, how do the amino acids join together? So I'm going to use an example for this, but again, let's just draw a general amino acid next to each other. So the way the amino acids join together is by forming the amide bond, also known as the peptide bond. And the amide bond, if I just draw it to the right here, now the way that's formed is by removing a water. And the water is always removed in the same way. You take off the OH, this is what I'm underlining here in green, you take off the OH from the carboxylic acid and the H from the nitrogen. And then that is ejected as a water, and then this bond is formed in the middle. So I think this is best demonstrated with an actual example from a past paper. And what I've done, I've deliberately picked an example here, which is just not a very nice one. And that's proline, because often when students see a ring structure, they think, oh, wow, what have I got to do here? It doesn't look very nice to deal with. So let's first discuss what are the 2 possibilities of these amino acids joining together. So we could have proline on the left joining to alanine on the right, or we could have alanine on the left joining to proline on the right. These different arrangements lead to what is called different sequences of amino acids, and then that can create different proteins. So especially if you're studying biology and biochemistry, this is done a lot in protein synthesis. So just the different order of amino acids creates different proteins. So it does matter how they join. So let's look at each one in turn, because the drawings are going to look slightly different. So we have then on the other side, now let's go ahead and remove the water. So I'm going to circle in blue the bits that are relevant. So remember what I just said. You take the OH of the carboxylic acid and you take the H of the NH2. Gives us, in the middle, C double bond O. Then that line there, which I'm just pressing in there, is now going to join directly to the nitrogen. And this hydrogen here with the dot is that one there. Now let's draw in the rest of the structure. So that is proline on the left and alanine on the right. So now let's look at the other way. Just draw here. So I'm going to do exactly the same thing again. I'm going to take off the OH of the carboxylic acid. And the only H to take off this time is this one, isn't it? There isn't any other one, because this nitrogen is bonded to a carbon on that side and a carbon on that side. So it only has one hydrogen. So this then is going to lead us to this picture. And the nitrogen is just going to be obviously still bonded in its ring structure. But the difference is between this bonding here and the one we just did a moment ago is, if you look at this amide bond here, that's got the NH in place. But in this case, the hydrogen is gone because nitrogen is now saturated. It has 3 covalent bonds and then a lone pair remaining, just like it would have a lone pair remaining here. But it kind of looks a bit strange, doesn't it? Because it's missing that hydrogen, but that is the part that would bond. So the hydrogen isn't there because this would be classed as a secondary amine, whereas in this case where you have just NH2 bonded to one other carbon, the classification for that is a primary amine. If you have any questions on this, please comment down below and I will get back and help you. I appreciate this seems quite technical. And check out the other videos I've made on this topic. I'm sure you're going to find some value in them. See you in the next one.